Gymnosporangium juniperi-virginianae is a plant pathogen that causes cedar-apple rust. In virtually any location where apples are crabapples and eastern red-cedar coexist, cedar-apple rust can be a destructive or disfiguring disease on both the apples and cedars. Quince and hawthorn can substitute for the apples as hosts, and many species of juniper can substitute for the eastern red cedars. Symptoms: on the apple tree, the infections occur on leaves, fruit and young twigs. The brightly colored spots produced on the leaves make it easy to identify. Small, pale yellow spots appear on the upper surfaces of the leaves, usually during late April or May on the eastern seaboard of the United States. These spots gradually enlarge and turn orange or red and may show concentric rings of color. Drops of orange liquid may be visible on the spots. Later in the season, black dots appear on the orange spots on the upper leaf surface. In late summer, tube-like structures develop on the undersurface of the apple leaf. Infected leaves sometimes drop prematurely, particularly during drought conditions or when the tree is under additional stress. Infections on fruit are usually near the blossom end and are somewhat similar to the leaf lesions. On the eastern red cedar host, the fungus produces reddish-brown galls from 1/4 to 1 inch in diameter. These galls can be mistaken for cone structures by the uninitiated. After reaching a diameter of about 1/2 inch, they show many small circular depressions. In the center of each depression is a small, pimple-like structure. In the spring, these structures elongate into orange gelatinous protrusions or horns. The spore-bearing horns swell during rainy periods in April and May. The wind carries the microscopic spores to infect apple leaves, fruit and young twigs on trees within a radius of several miles of the infected tree. On other species of juniper more common in landscaping and bonsai, the sizes of the infections are reduced. Early in the infection, the galls are small bumps on the woody portions of the plant. They maintain the orange gelatinous form after the first warm rains of spring, but generally on a greatly reduced scale. The disease cycle. Understanding of the disease cycle of this rust fungus is necessary for proper identification and control. Cedar apple rust is caused by the fungi Gymnosporangium, or more specifically Gymnosporangium juniperi-virginianae, that spend part of their life cycles on eastern red cedars growing near orchards. The complex disease cycle of cedar apple rust, alternating between two host plants, was first delineated by Anders Sandow Orsted on the eastern seaboard of the United States. At the first warm rain of spring, the spore horns become gelatinous masses and produce the teleospores. Wind carries the spores to apple leaves at about the time that apple buds are in the pink or early blossom stage. Upon reaching apple buds leaves, the spores attach themselves to the young leaves, germinate, and enter the leaf or fruit tissues. Infection takes place in as little as 4 hours under favorable conditions. Yellow lesions develop in 1 to 3 weeks. In July and August, spores from the apple leaves are produced. The wind carries the spores back to eastern red cedars, completing the infectious cycle. The spores land on cedar needle bases or in cracks or crevices of twigs. There they germinate and producing small, green-brown swellings about the size of a pea. Galls do not produce spores until the second spring. However, mature galls usually are present every year. This fungus produces 4 out of 5 of the spores known to be produced by the class Erdinomycetes during its life cycle. Rust fungi have a complicated life cycle with up to 5 types of spores in its life cycle, and often an alternate host, and an alternate host as well. The Erdinomycetes that have all 5 spore stages and those with less are said to be macrocyclic or microcyclic, respectively. Control. Because apples are an economically important crop, control is usually focused there. Interruption of the disease cycle is the only effective method for control of the cedar apple rust. The recommended method of control is to remove cedars located within a 1-mile radius of the apples to interrupt the disease cycle, though this method is seldom practical. For those doing bonsai, it is common to have the trees within feet of each other and on the central eastern seaboard of the United States. Eastern red cedar is a common first-growth conifer along roadsides. There are differences in the susceptibility of various apple varieties. Jonathan, Rome Beauty, Wealthy, and York Imperial, US susceptible. Grimes, Golden, Narragansett, Red Delicious, Winnesap, Stamens, Red Free, Juniper, and Priscilla are resistant. Crabapples are generally more susceptible than apples. Resistant crabapples include Adams's, Beverly, Candied Apple, Dilgo, Donald Wyman, Ellie Inglis's, Indian Summer, Lucette, Mount Arbor, Empacisifolia, Red Jewel, Robinson, Robusta, Royalty, M. Sargentina, Snowdrift, and Special Radiant. Resistant crater eggs include C. crossgalli, Ceres intricata, C. levigata, Autumn Glory, C. phenopyrium, C. pruinosa, C. Varieties, and Winter King. The resistant varieties are less susceptible to attack, but that does not mean that they are free from an aggressive attack. Fungicide sprays applied in a timely manner are highly effective against the rust diseases during the apple cycle. Most sprays are applied 4 times at 7 to 10 day intervals, starting with pink bud on crab apples. These applications are to protect the apples from spores being released from the cedar host in mid-spring. If cedar apple rust disease is diagnosed on apple fruits and leaves, it is far too late to spray. Systemic fungicides are available. Fungicides listed for use on apples can be used in July and August on the cedars to reduce infection. Application of fungicides to the junipers before and while they are in the infectious orange gelatinous state seems to reduce the severity of the outbreak. Gallery.